Going on to lecture objective 4, describe the propagation of action potentials from the axon hillock to the axon terminal, and then compare propagation in myelinated versus unmyelinated axons. Now, if we think about the conduction of the nerve impulse, if we again take a step back and look at the entire neuron and some of the events we described earlier, the action potential is going to begin at the soma, or excuse me, the graded potentials, right? I said that incorrectly. The graded potentials will be picked up on the soma. The action potential will begin at the axon hillock, all right? And so this is why it's important to remember that the voltage-gated sodium channels are located on the axon hillock as well as the axon. So this is the length of the neuron where we have these sodium channels that will allow the propagation of the action potential down the length of the neuron. And so the action potential occurring at one place on the neuron allows for the depolarization of the adjacent neuron, right? This is going to help the action potential move in one direction. And so when you think about that, we're going to depolarize this part of the neuron. That's going to allow the excitation to spread to the adjacent neuron. And then because the neuron that is proximal, like because the part of the neuron that is on the proximal part, that is in its refractory period. So it's impossible to fire another action potential on this part of the membrane, on this part of the neuron membrane. This is what prevents the action potential from moving backwards. All right, so the refractory periods are important not just for keeping the action potential confined to the same duration and the same length and the same amplitude, but it is also preventing the retrograde movement of the action potential by temporarily inactivating the segment of the neuron that has just undergone depolarization. All right, so the depolarization will propagate in one direction only. And then after this second segment of the neuron becomes repolarized and it is in its refractory period, then the proximal part will then become resting, right, in the resting memory potential, and then another action potential can subsequently depolarize this proximal segment. All right. So that is how the action potential jumps down each segment, right? Allowing that proximal segment to stay repolarized and then allowing the depolarization to move in one direction. Now, the conduction rate in some neurons is going to be slow, simply because the action potentials are generated by each individual event. And so the amplitude of each action potential will be the same, without any loss in that, in the strength of that signal, if that makes sense. So this is another reason why it's so important for each action potential to be identical. So we don't lose the strength of the signal. We don't lose or dissipate the strength of that signal or the strength of that potential as it moves down the length of the neuron, right? Which we know is true for our graded potentials. So here is our membrane. It's currently at rest, right? Inside is more negative than the outside. Here is site A with the original location of the action potential. That then propagates to site B, which is still at rest. And then we can see site B is now depolarized, and that can no longer depolarize site A, but it can depolarize site C, and that is because site A is still in its refractory period, and so it cannot be depolarized even if another graded potential came along, right, which is the case with this site B. Then we move on to site C becoming depolarized. Site A is back in the resting state, and then site B is now in the refractory state. And so the action potential can only move from site C to site B. All right. So that is how the action potential moves down the length of the axon. This is all kind of repeating those same events. Now we want to describe the way that conduction happens in a myelinated neuron. So typically our autonomic neurons, for example, the neurons that control our smooth muscle, our glands, our cardiac tissue, those are not myelinated or not heavily myelinated. The neurons that are myelinated are going to be your peripheral neurons, right? Especially your skeletal muscle neurons. So your radial nerve, for example, I'm sure you guys have talked about radial nerve in your labs, right? Your radial nerve, excuse me, your median nerve, your ulnar nerve, your sciatic nerve, those really large nerves that are responsible for propagating signals for long durations, right? Look at the length of your limb, right? The length of your upper limb, the length of your lower limb. These neurons have to send signals along the entire duration for within a short period of time. So those neurons are going to be more heavily myelinated. And what that myelin does is it improves, excuse me, or increases the speed of that propagation, the speed of that action potential traveling down the length of the neuron. So there are spots on the neuron called the nodes of Ranvier, and the nodes of Ranvier allow sodium and potassium to cross the membrane every 1 to 2 millimeters. So these nodes are basically gaps in the myelin, all right? And what this does is that the action potential can kind of leapfrog over certain segments of the neuron. So it doesn't have to go from site to site to site, right? It doesn't have to go sequentially, depolarizing every single segment. It can kind of just leap over a bunch of segments, right? And one of the best examples I have for this is I used to live in New York, and there were local trains that would stop at every single station. And then there were express trains that would just go across maybe 5 or 6 stations. And so those would move very, very quickly, particularly for folks who had to go very far in a short distance of time. So a typical unmyelinated neuron is like the local subway. It's going to go from every single spot, stop at every single station. And then the myelinated neurons have this ability to sort of run express, right? They're going to skip a bunch of stops and kind of just leapfrog down the length of the neuron. And so this allows the action potential to undergo what's called saltatory conduction. Saltatory conduction means that it's not going to every single site on the neuron. It's going to just leap over these myelinated segments. So these segments that are covered with myelin are going to really act as an insulator. And if you think about your cables, right, your charging cable, your electrical cables in your home, those act in the same way. Insulating that wire actually speeds up the conduction as it moves down the length of this neuron, right? Okay, another example of that here. So here is what a myelinated neuron looks like. And we'll talk about what myelin is coming up in chapter 9. But myelin is basically fat, right? It's basically fat that wraps around these intervals along the neuron and kind of just protect that spot from having to be depolarized so that the action potential can leap right over it, right? So that's all that myelin is. So notice how this is the depolarized segment here. It's going to skip entirely these middle segments. And then the next segment to be depolarized is this other gap here in the myelination, which we refer to as the node of Ranvier. All right, so these openings where there are no myelin or there is no myelin present, those will be the nodes of Ranvier. And these are the only segments of myelinated neurons that actually undergo depolarization. Okay, last little thing we want to say here is kind of thinking about how the action potential speed differs depending on the diameter of the neuron and the type of neuron that it is. So the bigger diameter neurons are going to have an increased speed, right? This is why, again, neurons of your peripheral limbs are thicker, they're bigger. The sciatic nerve, for example, is the thickest nerve in the body. It has to go along the length of your entire lower extremity, literally from your pelvis where it starts all the way down to the tips of your toes. So your sciatic nerve is really thick. It has a really large diameter. It's also very heavily myelinated to allow for that, speedy, high velocity of the conduction. Whereas thin neurons, unmyelinated neurons, are very small and they're going to control different types of signal. So not necessarily your muscle or your sensory perception, or even your movement, but really smaller, really other functions like your autonomic function, like your heart rate, like your glandular secretion, your smooth muscle, the innervation to your gastrointestinal tract. Those neurons are not myelinated. They're really, really small. If you were to look at your cadaver dissection and go into the abdomen and look at some of the neurons that innervate your gastrointestinal tract, look at some of the plexuses that innervate your kidneys, your liver, your spleen, they're really, really tiny neurons, almost invisible to the naked eye, and they're not myelinated, right? And that is because they don't have to send signals along as long of a distance as your peripheral neurons do. And they also don't have to be as fast. If you think about the speed of reaction that you need for your peripheral muscles, for your sensory perception, for your motor function, that needs to be rapid, that needs to be fast. When I think to move my hand, that needs to be immediate. If I touch a hot plate, that reaction needs to be spontaneous, it needs to happen right away. So these neurons need to be much faster than, say, the absorption or the stimulation to your gut as food is moving through. That doesn't have to be as reactive, as responsive. So that is another reason why your peripheral neurons have to be heavier myelinated, have to be thicker, whereas your autonomic neurons can be much smaller and not necessarily myelinated.